Okay, thank you. First of all, thank the committee organized the seminar series and invited me again. Last time I talked about the synthesis of a lipid nanotube for the controlled drug delivery and the light harvesting. So for this presentation, I will focus on the engineering of a liquid crystal droplet for the biosensing application. As you know, liquid crystal is the phase between the crystal and the liquid. And in the liquid crystal phase, and the molecule show the long-ranging orientation order, but does not show any position orders. So, and the liquid crystal has been widely used in the display industry based on the how liquid crystal orientation changes response to magnetic field and electric field. So we also know the liquid crystal orientation is extremely sensitive to the surface which liquid crystal in contact with. And we have been used liquid crystal as an optical probe to detect the, the ordering of the molecules in the lipid membrane domains. You can see here the number of the two pictures and show the orientation order of the molecules in the lipid domain. Due to the orientation correlation and the surface-induced or lipid molecule-induced ordering of liquid crystal can be amplified into the bulk due to the long-range orientation order. What we find out is that, they have a membrane, have a thickness 1 or 2 nanometers after liquid crystal amplification, and the ordering induced by the lipid molecule can amplify into a liquid crystal phase through the distance around 6 microns. So, and for this talk, I will focus on the design of liquid crystal droplets and use them to serve as the simple and the fast and the sensor platform to detect the important biological species. Why we interested in liquid crystal droplets? You can see here, this is the polarized microscope image and show the liquid crystal droplets can adopt a different type optical patterns, depending on how the liquid crystal oriented within the droplet. For example, in this case, liquid crystal molecule oriented around the surface of the droplet have 2 defects point. So in this case, we call the pre-radial direct configuration, and the liquid crystal oriented around the line I'm showing here have 1 defect. When the defects move to the center of the droplets, liquid crystal show the perfect radial direct configuration, all the molecule oriented around the radial direction. So the orientation of the liquid crystal inside of the droplets actually depend on the surface anchoring energy. So when the molecules that we interested detect absorb on the surface or jump out from surface, will trigger the transition from one type configuration to the other type of configuration. When, in this case, when the beta CD surfactant complex jump to the surface or absorb on the surface, can trigger the transition of a configuration of liquid crystal droplets from bipolar to the radial. In the presence of the molecule we internally detect, we are competing with the molecule we pre-put on the surface of the droplet, can remove the surfactant from surface, trigger the transition. These direct configuration changes can be easily to observed using ordinary microscope. So this is the concept to design the liquid crystal droplet based on biosensors. So now the first issue, the key issue is we need to first stabilize the liquid crystal droplet, generate a predetermined direct configuration, then use that as the sensor probe to detect important biological species. So in this talk, I will show 3 or 4 examples how to use the liquid crystal droplets to detect the biomarker, virus, and the pathogen. Now, the first issue is to stabilize liquid crystal droplet. and induce the very precise direct configuration. So we have been used so-called surfactant and the lipid to stabilize liquid crystal droplets, induce the so-called radial direct configuration of the droplets to detect small biomarkers. And we also use the biopolymers to stabilize the liquid crystal droplets, induce bipolar direct configuration of the droplets, and to detect the protein, virus, and the cancer cell. As well as we use peptide to engineer the surface of the droplets, induce a radial direct configuration for detect bacteria. Now, the first project I have been working on for a while is, to engineer the surface of the liquid crystal droplets to detect the biomarker for colon cancer. We know the bile acid is a biological surfactant, which plays central role for us to digest the fat. And due to the surface activity or self-organization capability, bile acid able to pull the fat molecule out of the bulk fat from a micelle, water-soluble micelle, for cell uptaking, provide us the nutrition. However, the secondary bile acid I'm showing here, called the LCA, and is overexpressed when the individual person have a colon cancer. Okay, so compared to the LCA, the secondary bile acid, they only have one hydroxy group. The other bile acid also present in the liver, and they contain 2 or 3 hydroxy groups. It's extremely to distinguish LCA from other bile acid in the biological flow system. So we're interested in detect the LCA because this is the cancer mark. So first of all, we engineer the surface, use the biopolymer, we call the hydrogen, and use the emulsion method, allow the polymer dissolve on the surface, stabilize the droplets, and then trigger the gelation and mobilize the liquid crystal droplets inside of the hydrogel. Hydrogel contains the water and small molecule like a surfactant can penetrate into or can diffuse into the hydrogel, dissolve on the surface, generate radial directed configuration. So these changes can be easily observed by using polarized microscope. In the before adsorption of the surfactant, you can see the ring-like pattern. After absorption of surfactant, direct configuration of liquid crystal changes to the radius, show the cross patterns, provide a simple optical signal for us to detect the molecule we're interested. And so for the detection of LCA, bile acid, and we place the hydrogel, which contain surfactant stabilized droplets, into the glass dishes. You can see before we add the LCA, bile acid, the droplets mobilized in the network show the cross patterns. After we adding the LCA, you can see the changes, and from the radius to the pre-radius and to the bipolars show the ring-like patterns. So that's the signal we're used to detect the LCA. The detection mechanism is based on, as I showed in the previous slide, LCA are very surface active, very hydrophobic. It can penetrate into the, diffuse into the hydrogel, reach to the surface, remove the surfactant, trigger the transition. So here is my transition curve of the emburied liquid crystal droplet in the hydrogel from the radius to the biopolars. You can see here, only LCA able to trigger the transition from the radius to the bipolar. CA and the DCA, which is coexisted with LCA, are unable to trigger the transition from radial to the bipolar. So in this case, we can use the liquid crystal droplets embedded in the hydrogel to selectively detect the LCA. So by changing the number of the droplets, you can see my transition curve shifted from the right to the left, which indicate my detection limit are reduced when we reduce the number of the droplets embedded in the hydrogel. So in this way, we can control the sensitivity by reduce the number of the droplets embedded in the hydrogel. We can change the detection limit from the around 70 micron to the 0.1, 0.2 micron. So we also can use the droplets to detect the LCA in the different biological fluid, such as urine, synthetic urine, a PBS solution. Now I'm interested to detect the biomarker. A bioassay here is the LCA from the biological fluid and the cancer patients. I have not found the collaborate to provide me the biological fluid from the individual person with the colon cancer. So that's my interesting. After I prove the concept, show the sensitivity, we can use the droplets for clinical diagnosis of the colon cancer. And the second project I work on is engineering liquid crystal droplets to detect the virus. The one virus I work on is called the Copy mosaic virus. Diameter is around 30 nanometer. You cannot easy to see, but the surface is highly inactive charge. To detect the virus, and we use the emulsion method to generate the liquid crystal droplet. In this case, the biopolymer, we call the PEI, a positive charge, can absorb on the surface and provide the positive charge surface. Same time, induce the bipolar direct configuration. So in order to observe, in order to prevent the diffusion of the droplets out of the focal plane, when we use the microscope to monitor the transition of direct configuration of liquid crystal droplets, we designed the microfluidic channels, have very, very thin channels. So the droplets cannot diffuse out of the focal plane. You can see here, before I add the virus, the droplets show the ring-like pattern, means that the molecule adopt radio direct configuration, just like cartoon showing here, along the surface, oriented around the surface. After I add 50 micromole virus into the channel, you can see over time, and the transition take place, and the transition completely was in around 2 minutes. So the transition mechanism is a result of the electrostatic interaction. We know the virus is an active charge. Electrostatic interaction will attract the virus to the surface of the droplet. Now I generate virus is negative. I generate a polar shear around the liquid crystal droplet. Liquid crystal contain very large dipole here. So dipole-dipole moment interaction is the result of the transition of the electron configuration of the droplets. Now, so we can see the sensor, this is my transition curve. We are able to detect the virus with the concentration around the 25 micron. And we also tried other virus. The virus I'm working on is non-toxic. So the important to detect, for example, COVID or the other toxic virus. But I do not. My lab is not allowed me to do that. So the project I want to talk about is to detect the bacteria. Okay, so first for trying to engineer the surface of liquid crystal droplets. So we know that antimicrobial peptide, this is the chemical structure of the peptide. The peptide can adopt different configuration or conformations. So one conformation is alpha conformation. Alpha conformation of the peptide show the strong bonding affinity to the bacteria and can penetrate into the wall of the bacteria, eventually kill the bacteria. So use the antimicrobial peptide, we stabilize the surface of the droplet. same time induce the radio-directed configuration. So then we add the droplets into the solution which contain E. coli, non-passive genetic virus, non-toxic virus. You can see here is the microscope image. This is my droplet of the liquid crystal. And this is the E. coli. Okay, so corresponding the polarized microscope image show the droplets show the ring-like pattern, means that bipolar directed configuration in the presence of the E. coli. So we know the peptide shows strong bonding affinity to the bacteria. So in the presence of the bacteria, the peptide initially adsorbed on the surface of the droplet is going to jump off and jump onto the virus. Same time, trigger the transition of liquid crystal from the radio to the bipolar, provide the simple signal to detect E. coli. So then we manipulate or we reduce the concentration of the E. coli, trying to figure out the detection limit. You can see here the transition happened around the 100 CFU, that's the unit, allowed us to count the number of the virus in the solution. So this is a very, very low concentration of the virus can be detected by using the peptide functionized liquid crystal droplet. We also tried a different type, green negative, green positive virus. And you can see this is my transition curve. Detection limit is around 100 CFU, roughly 2 nanomole, a very, very low concentration. So now I'm also interested in finding collaborators to see whether we can use, after we prove the concept and figure out the detection limit, figure out the detection mechanism. So see whether we can detect the passenger. So now I'm going to wrap up my presentation. First of all, I'd like to thank the students and did all the work. Mainly the work done is by Temme and Jenna. So the virus, COVID mosaic virus, CPMV, is provided by the Dr. Wang group from the USC. And the peptide was synthesized in the Dr. Guy's group at the UPenn. I received 3 NSF award on this topic, published the 26th journal paper. Thank you, and happy to answer the question you might have.